Hello, Morgan. Susan. So, welcome. I don't think you were here last week, Susan. How are things? How are things going with you?
Well, I've tried several different models, and none of them work. They seem to don't quite resolve. I can't get the bottom part out of the red, so to speak, no matter what I do.
Yeah.
Huh. Like I said, I think it thinks that I'm building a bridge or some sort of large structure, and I'm not.
Oh.
Cells float in water. Their parts are they float in water, so they're not pressing down on the bottom. They don't do that. So I'm having some issues with it. It's just lovely pictures with the bottom red. And the only way I can get the images of these things is to what they say is called write a report.
Oh.
So it doesn't turn up until I say report. And so I have, over a dozen reports with different variables to try to fix this. And no, this is still red. So I'm meeting with the professors this afternoon, and I'm going to ask the one fellow who knows something about mechanics, what? Yeah. I'm an electrical engineer. I've just been trying to do this program, like, I don't know AutoCAD really. I don't know. That's the only thing I might know. And this console thing is just got the better of me, so I'm just going to write it up, okay?
So that's using COMSOL.
They want me to do that. Pardon?
That's using COMSOL?
Yeah.
All right.
It's very involved. Like, I wanted to find the middle of a line, so I have to kind of break up the line. That's the easy way to find the middle of the line. And then I need it back together again. Like, I can't have it split and operating independently, or my structure doesn't work. How do you do that? So, well, I did try to combine it, but it's still not working. So I don't know. It's like, I have no clue.
Right.
If you can see behind me, I have a bunch of stuff I need to sort through, finish writing it up.
Yeah. Well, good luck with that. Yeah. Sorry that COMSOL didn't work out. I don't know.
Yeah, I've talked to students about it and other people, and they just don't like it.
Right. Oh, yeah. Well. How about some of the other programs, like we talked about, Morpho and some of those? Are those like still on the table? Or?
I wanted to try Morpho, but I couldn't get it downloaded on this computer. The last line, like it's Ubuntu by line program. And I'm willing to try it, but I've never really used something like that before.
Yeah. Well, yeah.
And my professor says, no, just use MATLAB. We just want you to use MATLAB. And I'm going, well, I wish I'd used MATLAB before. Like I could suggest this, but.
Right.
So, and MATLAB, you can easily transfer it to Python if you need a programming language, although
right.
It's related. Anyway. And MATLAB's nice. It gives you an error message. You take the error message, you copy and paste it into Google, and it comes up with all these people who solved this error message, and they did this and that and the other thing. And you go there, oh, that one. And you pluck it out and put it in your program, and it works.
Yeah.
It's just like, you look up an error message in console, it refers you to the manual. You look in the manual and it's thick.
Yeah.
Or it's in computer ease or something.
Yeah.
And you're trying, okay, well, and what on earth does that mean? It takes more than an hour to read a page because you're going, yeah.
Great. Yeah.
Anyway, this is my complaint. It's hard to figure out error messages in console, or like what problem I'm having. I don't know. I sent it into the help desk and they get back to me a couple of weeks later.
Yeah.
No, that's not a help desk. Sorry. It's just a tease.
Yeah.
Anyway, so how are things?
Okay. Yeah, we're just rolling along, sort of at the end of the semester, and hopefully things end without too much incident. So, and for the term, for the year.
Yeah. That's why I want this written up. I want it written up for the end of the term, and my PowerPoint started for presentation, and just do it.
Yeah.
Anyway, yeah. So I've tried 4 or 5 configurations of this, so that should be enough.
Yeah.
They all don't work in some way or other. Yeah. There's a phone call.
Yeah.
Good. And my phone has been having trouble.
Oh, yeah. Okay. So I guess I can talk about a few things. Yeah, there's this. Let me share my screen.
Oh yeah, I have a paper I should send you.
Oh yeah, what is it?
Try to remember.
Okay. So I just wanted to point out. Well, I did talk about the graph learning on graphs conference, and I have yet to summarize it reasonably, but it just I've been going through the materials, but I haven't been able to summarize it. So I'll do that later, maybe by the end of the year. We'll see. It's a lot of stuff. So it's like, last year they did. They kind of went over the basics of the field, and the field is moving pretty fast. So there are all sorts of different approaches that people are using. One of the newer approaches, of course, they're focusing on topological methods, things like that. They're also focusing on what I think we talked last year about different ways that you define a connection between two nodes. And they do a lot of things with message passing to sort of extract the networks and to build networks from data. So there are different methods you can pull in from communication networks to help. And as for the types of networks that they had, a lot of them were biology. Some of them were proteomic. There were some applications to medicine. There were also a lot of data science techniques. So analyzing large data sets with these kind of methods. So it's not just theory that they had. They had a lot of stuff.
Anything on actual physical networks?
They did have a lecture and a workshop on physics in physics, but I don't know if it would have been particularly relevant to what you were doing. So
Okay,
yeah.
I'm supposed to take these models large and figure out what it would be in a large tissue.
Okay.
And I think I'm going to go for another approach rather than building the tensegrity to encompass everything, because there's the vertex model of cells uses just the shape of the top apex of the cell to determine its behavior. And so one might be able to just work with a simplified model to get most of the behavior of the cell.
Yeah.
Yeah, just I'm kind of fed up with trying to get tensegrity to work.
So you'll try other approaches if you have to. Yeah.
Oh yeah. No, I've already looked at. I've what did I say? Epithelial cells are modifying Kongami lattice. That was it.
Okay. Yeah.
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 Anyway.
So can I get? Oh, go ahead.
I'll send you this paper later. I found one on C.elegans, I think it was, and then there was another one I thought you might like.
Okay, yeah, that'd be great.
Okay, I'll turn off my camera so that people don't have to look at my mess. Behind me.
Yeah, so
I'll straighten it up.
Oh yeah.
I hope I can just write this up and straighten up my mess.
Yeah. All right. Yeah, so that's learning on graphs. There's this other event coming up early next year. This is the Virtual Cell and Development Festival Week. And this is something I'm going to submit a abstract for. I have to submit it by January 5, so I might talk about it next week. We have a lot of things in the group that we could submit to this. It just has a lot of different things that you can, different mini symposia talks, professional development plenaries, and other types of events. So this is something that, might be of, I'll see what kind of what I think about might be appropriate for this. This is like 15-minute talks, so.
So would my tensegrity of cells fit in with that?
Oh yeah, probably that would work. I mean, they have I guess it's pretty diverse what they're looking for. So this is just a virtual thing, so it's like you can participate from anywhere in the world. So, and it's a wide variety of topics in cell and developmental biology. So it's the SMB subgroup on cell and developmental biology. And so this is the Society for Mathematical Biology. So they're, that's exactly what they're interested in, that type of thing.
Okay.
So, yeah, I can send you a link to the thing if you want.
Oh, okay.
Okay. So yeah, that's that. And then we've had some other types of things going on in the Slack. So Jia Hong sent this paper along. This is an older paper from 2018, but it's actually Zhirong Bao, who, of course, has been involved in a number of efforts to get cell data for C.elegans embryos made public, and he's working with a number of people here on this paper. It's called Deep Reinforcement Learning of Cell Movements in the Early Stage of C.elegans Embryogenesis. So I'm going to go through this paper, and this is basically where they're using agent-based models, and they're using deep reinforcement learning along with that to analyze some of the 3D time-lapse microscopy images that we talk about a lot in the group. So I don't know if there was something in the chat here. Oh yeah, I'll send the link to the virtual cell meeting. So I just put that in the chat for Susan and Morgan, this link. Okay.
Copy.
Yeah. So yeah, this is let's look at this paper a little bit and see what they're doing, what kinds of techniques they're using, and what their results are. So this was published in Bioinformatics journal. So the motivation is is that cell movements in the early phase of C.elegans development is regulated by a highly complex process in which a set of rules and connections are formulated at distinct scales. Previous efforts have demonstrated that agent-based multi-scale modeling systems can integrate both physical and biological rules. And so, you can have those physical rules of interaction, but also the biological rules that, we've talked about, and provide new avenues to study developmental systems. However, the application of these systems to model cell movement is still challenging. It requires a comprehensive understanding of regulatory networks at the right scales. And so, deciding what those rules are, is it are they force interactions, or are they, different types of molecular signaling, or what is going on there? What are the rules that we need? And remember, we don't need to take every single factor into account. We just need to abstract that to some key factors that produce something that looks like C.elegans, and then work from there. So if you have, physical rules that are very bare bones, it's okay as long as it produces a good result. However, the applications of these systems to model cell movement is still challenging and requires a comprehensive understanding of regulatory networks at the right scales. Recent developments in deep learning and reinforcement learning provide an unprecedented opportunity to explore cell movement using 3D time-lapse microscopy images, which is what Jirong Bao does a lot of. He's got a lot of experience in acquiring those and analyzing those for, different types of molecular signals. So in this paper, they present a deep reinforcement learning approach within an agent-based modeling system to characterize cell movement in the embryonic development of C.elegans. So this is where they're characterizing the migration of cells after division. They're moving around the embryo, kind of acquiring new positions. So if a cell divides, the daughter cells will move to the left hemisphere and the right hemisphere. Or you'll get cells that migrate from one part of the connectome to another region. The shape changes, those involve a lot of movements. We want to be able to characterize those. But of course, those all depend on rules between cells interacting in space. Our modeling system captures the complexity of cell movement patterns in the embryo and overcomes the local optimization problem encountered by traditional rule-based, agent-based modeling that uses greedy algorithms. So there are these. This is the type of agent-based modeling that we use, where we just simply, use a set of rules. We have a set of cells that are fixed in space, and they just interact based on the state of their neighbors. And so, or you'll have some sort of, contents of the cell that have to be optimized in some way. So.
Greedy algorithms, that kind of goes with robotics. Like it's based on Boltzmann's constant, and I believe in robots. Exploration of the environment works better with things that are not greedy, like it.
Yeah.
Anyway,
yeah.
That's where I ran into that, was a course on robotics.
Right. Well, it's one application domain. So yeah, it's just the way they're optimizing the problem, and they're kind of considering other types of algorithms for some of these movements. Yeah. We tested our model with two real developmental processes: the anterior movement of the CPAAA cell, which is in that C sublineage. It's posterior, and then it's anterior, anterior, anterior. So like the divisions in C.elegans are often like in these sublineages, and they have their sort of the nomenclature talks about or specifies the direction in which the division occurs. So there's a posterior cell and an anterior cell in C. And then there are three anterior divisions. So when you have a division which cell is anterior, which cell is posterior. So you get this one division that's towards the tail and 3 divisions that go towards the head. And then they migrate based on that sort of orientation. So that's why they have those kind of names. But basically, this cell is moving anteriorly by intercalation, which means it's moving in between other cells, and the rearrangement of the superficial left-right asymmetry. So there's this superficial left-right asymmetry that gets rearranged as the cell is migrating, and so they look at that. So they have basically what they have to do is they have to explore their environment. They have to have the cell explore the space in between other cells, so it has to move through the embryo space. It has to sort of squeeze in between other cells and find its adult position. And so this is where things are getting rearranged because you're moving cells around. In the first case, the model results suggested that CPAAA's intercalation is an active directional cell movement caused by the continuous effects from a longer distance or farther than the length of 2 adjacent cells, as opposed to a passive movement caused by neighbor cell movements. So this is where they kind of, if you have cells that are next to one another, they're kind of moving around and you're getting kind of squeezed or moved around as a result of that. And so this is like where there are adjustments that need to be made based on what your neighbors are doing. And that would be like typical of a typical agent-based modeling sort of setting where you're basically responding to your neighbor's states. But in this case, you're looking at these longer distance active movements, which involve the cell actually moving to a new location, acquiring new neighbors, and rearranging the grid that this process is being played out on. So these results showed that our approach to introduce deep reinforcement learning into the agent-based modeling paradigm can test regulatory mechanisms by exploring cell migration paths in a reverse engineering perspective. This model opens new doors to explore the large data sets generated by live imaging. So this is the whole point of this, was to sort of have a model for live imaging data to sort of get some of this real-time process down. And, it gives us a little bit more flexible approach to this. So the source code is available here at this GitHub repository. If you look at the GitHub repository, we see that it's this is DRL for cell movement. It's just kind of hasn't really been developed that much since, but it's basically, I think it's supposed to be like a legacy repo. So this is the paper, link to the paper, and so basically tell you how to run it. You use PyPlot, PyTorch, NumPy, Mesa, and Python 3. So they're running this on Mesa, and basically it tells you how to use it and install it. So that's the repo. You have the model and then the run.dqn file. This is the Mesa model. Here you can see that you have all the different things. I don't, if we want to get into this code too much. But basically, they're modeling a series of cells. You can see here the state cell list. You have to define some of the parameters of the cells. You have to adjust the axes, and then you go through and you run the simulation where it's drawing the plane, it's drawing the grid for the agent-based model, and then it's running the program on that with these different parameters. And you have a grid full of cells, and it's calculating a radius around the cell to determine what its neighbors are. And I guess you're moving the cells around, and then you're seeing what the neighbors are. You're redrawing the radius. But it looks like a pretty good approach. It's using Mesa, and so let's look at the rest of the paper and see what people or what they've been able to do here. So so they have they talk a lot about the recordings. Talk about agent-based modeling and directional cell movement, which of course is what they're doing here. So directional cell movement is critical in many physiological processes during C.elegans development, including morphogenesis, structure restoration, and nervous system formation. It is known that in these processes, cell movements can be guided by gradients of various chemical signals, physical interactions at the cell-substrate interface, and other mechanisms. So this is there's a big literature on this, especially in C.elegans. It remains an open and interesting challenge as to what and how one could learn about the rules and mechanisms of cell movement from the movement tracks recorded in live imaging. So, when we have this live imaging, we have these movies, we have these movement tracks that you can extract. We've done this in some of the projects where people have extracted cell tracks from the data. It's basically where the cell moves over time. So in each frame, the cell moves its position, and you can calculate that track. That track then, when there's a division, there are new tracks that are generated, and you can actually, that's very different from the grid that we usually think of when we think of agent-based modeling. So, that's something that has to be incorporated. And so this paper presents a new approach, adopting deep reinforcement learning approaches within this agent-based modeling framework. And so this is where we differ from the greedy algorithm that we usually use. And so this is the section where they talk about this. Deep reinforcement learning is good at dealing with high dimensional inputs and can optimize complex policies over primitive actions. So this is where you have these policies that you have to select, and it's kind of like a policy for interaction. So you select a policy and you go with it. So for example, if you had a cell that had a policy for interacting with other cells, it would just be like a set of rules that it would adopt over another set of rules, and then would do that. It would execute on that. So this is a little bit different way of thinking about it. With a regular agent-based model, you just have a single set of rules you usually implement over the entire simulation. You just kind of choose your rules and say, this is what we're going to do. In this case, what they're saying is that we can actually create policies that allow us to select our rule sets, especially when we're moving around. When we move around, we have different rules in different parts of the embryo, or different rules over time. So this is where this naturally aligns with the complex cell movement patterns that occur during C.elegans embryogenesis. And so you can characterize this without knowing all the details of the biology. It just kind of, gives it some flexibility. And so we tested our model through two representative scenarios during C.elegans embryogenesis. The first is the anterior movement of CPAAA by intercalation, and then the rearrangement of the superficial left-right asymmetry. So these two cases where you have intercalation and this asymmetry rearrangement. In the first case, we proposed two hypotheses for the intercalation of CPAAA, and simulation results indicated that CPAAA experienced an active directional movement towards the anterior. So it's moving towards the head, which is caused by the continuous effects from a longer distance rather than a passive process which it is squeezed to the target locations by its neighbor's movement. So this is where, again, your neighbors are moving around, you're kind of squeezing on the cell, and it's pushing it in a new place, wherever it fits. For the alternate hypothesis, it's that the cell is actually moving somewhere, and it's kind of pushing past the rest of the cells. It's acquiring new neighbors and so forth. So that's how they kind of think about this. In the second case, the frequently occurring leader-follower mechanism was also supported by the simulation results of the asymmetry rearrangement. So there's this leader-follower mechanism that people, I guess, haven't really talked about it, but basically if you have cells migrating, you might have a leader and you might have a follower. So if you have two neighbors, one does something first and the other follows along. or one does an action first and then the second one is impacted by by that other cell. So
Yeah.
there are all sorts of leader-follower hype. Oh, go ahead.
Um, it is so much like the robotics course that I took.
Oh yeah, yeah.
Again, I guess it's based on that.
Yeah. Well, yeah, it is. And then, there's also this whole area of game theory that involves like first mover dynamics and leader follower mechanisms in that area.
Oh, really?
Yeah. So there are these Stackelberg games that basically remodel, and they do this. This is kind of from the economics literature, but it's like firms that sort of innovate first, and they come up with an innovation, and then the rest of the firms in the market will follow. Or if there's a leader in innovating some new invention, the rest of the firms will follow. And so that's in economics, but you can apply that to biology, where you have these kinds of uh, hierarchies of action, where if the leader does something, it constrains the followers to doing that thing. And so you can see that with a physical system of cells, where if you move into one space, the other cell can't move there. They have to sort of move maybe in or adapt their movements to tailor what the other cells have done before them. So it really constrains the embryo. We actually we've talked about this in a number of papers in our group. Actually, we have the paper on the cybernetic embryo from 2018, and then another paper from 2020 on connectome wiring using the leader-follower game or leader-follower dynamics. I'm glad to see that they're going in this direction as well. Okay, so in the meeting I mentioned two papers that the group has done on first mover or leader-follower dynamics. And so leader-follower dynamics, it's an emerging topic, I think, in cell biology, thinking about cells that are migrating, cells that have sort of moved first, and then cells that follow along. So leader-follower. Or you can have these first mover dynamics, which means that the cell that moves first, its preference, and the constraints are largely geometrical. This is something we can map from economics to cell biology. We have tools like Stackelberg equilibria and other types of tools in economics that we can transfer to cell biology as well. So that being said, we have a couple of papers that we've done in the group on this. One from 2018, which I am showing here, and one from 2020, which we'll talk about next. So this paper is in Biosystems. It's called Origins of the Embryo, Self-Organization through Cybernetic Regulation. So this is Robert Stone, Tom Portages, George Michalowski, and myself. Tom is a longer-term member of the group who doesn't attend much anymore, but he used to attend all the time early on in our group's history. Rob Stone is a collaborator. He wrote an original paper on what he called the cybernetic embryo, and then he wrote this as sort of the second version of it. And so actually we worked on this all together. We cover a number of things in this paper. We talk about sort of this idea of the cybernetic embryo, kind of develop some of the themes of the cybernetic embryo, some things about recursivity, some things about different types of complexity, talking a little bit about systems, and then getting into some of these other issues with respect to thermodynamics. One thing that we do talk about in here is this figure here, figure 4. So this is when we get into this discussion of first mover dynamics and embryogenesis. We can see that we can map embryogenesis, or very early embryogenesis, roughly to a game of tic-tac-toe. And you say, well, what does that have to do with anything? And the answer is, of course, is that tic-tac-toe has this first mover dynamics built into the game. So if I start a game of tic-tac-toe, I put an X in the middle, and then, in the center square. And then if you want to put an O, you have to play defense, put an O in the lower right square. And then my possible moves are to block you. So now I'm playing defense. But the first mover always has the advantage because they can set the terms of how the space is partitioned. What are the subsequent moves? And so this is a game of tic-tac-toe, and in England they call this noughts and crosses. And noughts and crosses is actually has a rich history in game theory and in artificial intelligence research. So look that up if you're interested. But we can map this game of tic-tac-toe or noughts and crosses to an embryo. And so you can see here at the bottom in B, we have this embryo that's dividing. So we have a single cell that's fertilized that divides into two cells. And then the first cell at the top, which would be the anterior pole, you would have a division event. And that division event then would constrain sort of the positions of the second mover. And this isn't considering migration. This is just considering, a static embryo. And migration would influence this model considerably, but probably not, because as we talked about in the meeting, when you make your move, you're sort of leading the rest of the cells. So if one divides first, and this is not a conscious process like tic-tac-toe, but it's just when the event occurs. So if cell 1 divides first, it has two cells. Those two cells have to be somewhere. They can select their position, and then 2 divides, and those cells can select their position, and so on and so forth. And so, the way we've proposed this, it can create asymmetries in the embryo structure. It can create other types of constraints on the positioning of the cells, the shape of the embryo, and the ability for cells to migrate. So this is a nice model. We haven't really developed it much more than this. And then later in the paper, we talk about some of the thermodynamic constraints that might be constraining this as well. So 2 years later, I wrote this paper alone. This is Raising the Connectome, the Emergence of Neuronal Activity and Behavior in C.elegans. So in this case, you're focusing more just exclusively on the connectome, the neural connectome. This was published in Frontiers in Cellular Neuroscience. And this is the section Cellular Neurophysiology. This was in a special issue on interplay between ion channels, the nervous system, and embryonic development. So we also talk about these first mover dynamics, and in this case, in terms of wiring up the connectome. So this is an example here in figure 2, and this is a sort of a model of connectome wiring as these cells are differentiating into neurons and they start making their connections to other neurons. So this is in C.elegans, where we have this model of connectogenesis, which is what we call it, where you have cells that divide, and then they keep dividing, and then they start to differentiate into, in this case, neuronal cells. These neuronal cells get turned on at different points in development. So these numbers represent minutes of development at which these cells become neurons or become neuron-like. And so the idea is that there are different sets of rules that are connecting them together. So which cells are most likely to connect to one another? And so we review in the paper that there's been some work on connected, how cells get connected, that there are these hypotheses about age first and other types of hypotheses. So the earliest cells start to connect and then the other cells join in, or if there are other more complex rules. And so, we kind of recast this question in terms of the first and second mover dynamics using different rules. So we have these different types of coupling rules, these different types of logic rules, and then, of course, influences of first and second mover dynamics. So this is, just a way to kind of understand connectogenesis based on the age of the cell, but also based on a data set of synaptic connections during development. So this is a good paper that lays out, and again, it only lays out briefly this idea of first mover dynamics, but it does recast this in terms of connecting different parts of the emerging connectome. Raising the Connectome, the Emergence of Neuronal Activity and Behavior in C.elegans, in Frontiers for Cellular Neuroscience, and Origins of the Embryo Self-Organization through Cybernetic Regulation in Biosystems. And I look forward maybe to doing another paper on leader-follower dynamics or first-mover dynamics, maybe next year. So if you're interested in collaborating, let us know. So this is where you have this mechanism leader-follower That was also supported by the simulation results of the asymmetry rearrangement. So this is good that we have this sort of asymmetry rearrangement being based on this leader-follower mechanism. In summary, this framework presents a reverse engineering perspective to investigate regulatory mechanisms behind certain developmental processes. By formulating a reward function as the representation of regulatory mechanisms, different hypotheses can be tested via reinforcement learning procedures. By comparing the extent of similarities between the simulation cell migration paths and the observation data, such hypotheses can be either supported or rejected, which can facilitate new explanations of certain cell movement behaviors. So this is definitely, I think, a really good paper for framing a lot of this. This is the ABM framework that they talk about, where they have the greedy algorithm. It's really where you have, actually, I don't know if this is their ABM framework or the general ABM framework, but this is where you start. You set up you initialize the environment. You hatch initial cells, which means they are populated in the simulation. So I don't know if they're actually simulating cell division directly, but they're just populating the ABM with cells. They're updating the global timer, so there's this timer that goes. Then is the ending condition met? Is it in its place that it needs to be? If not, then you select the next cell to act. You move to the next location. You have a division event. You hatch a new cell, which means it appears, or you don't hatch a new cell. And then you ask, have all cells acted? And if they haven't, then you go back and you select the next cell to act. Then if all cells have acted, then you update the global timer. You maybe select the next cell to act, or you say, is the ending condition met? And then if that's true, then you end the simulation. So you go through rounds of time where you have a bunch of initial cells that populate the environment. You check to see if they're in the right location. You update the time, you move them again, and then you hatch new cells when they need to be hatched. And then if all cells have acted, then you end the simulation. It's pretty simple. And then the reinforcement learning framework is a little bit different. This is the typical diagram for reinforcement learning, where you have the environment, which is the embryo. All the cells exist in that environment. You have a state and a reward. So the state function and the reward function, the state is whatever position the cell is in. And then if it's updated, you have a state t+1. The reward, of course, is the current reward. So if there's a reward to move, then that reward is met. If there's a reward to stay in place, the reward is met if it stays in place. And then if you have these leader-follower dynamics, the reward fluctuates based on the first mover of your neighbor, or if you're the first mover, and the reward differs there. And so then the agent is the cell, and there's an action taken on the environment, which is its movement and how it rearranges everything else, or it sets the reward, sort of sets the new set of rewards that are happening. So you have to select these policies that define the interaction rules based on the reward, based on the current state, and based on actions. So
what does the C in the CPAA mean?
Oh, it's just the nomenclature. So it's like the C sublineage. It's the and then it takes a couple of divisions. It divides anterior posterior. So the anterior cell is one sister or daughter cell. The posterior is another daughter cell. And then you get another division, which is anterior posterior, but this time the cell is anterior, so it's towards the head. And then there are three divisions towards the head. So it's like if you think about the C sublineage, it's dividing along two axes, left, right, and anterior posterior. And usually a lot of the divisions in the nomenclature anterior posterior. So you have, like, the idea is you have these different divisions over time. So the daughters move like towards the tail, towards the head, and then that's where they end up. That's where they divide to. And then they move in addition to that. So the movements are additional. and they move wherever they need to be for their final location. But you can see that just having the cell divisions like be spatially explicit, they sort of, set up this geometry where it's almost like soap bubbles that expand in different directions along different axes.
So the CPAAA describes like the daughter and the granddaughter and the great granddaughter.
Yeah,
or,
yeah.
Yeah, sort of. Okay.
Yeah. So yeah, that's and so then they have all these cells that they populate their simulation with. Let's look at this figure too. This is figure 4. This is where we have a specific evaluation step for a single action. So the way this works is that you have your cells, this nomenclature. This is AB, AR, PP, PAP. So this is where you have the AB sublineage, and then there's an anterior division, a division to the right, a division posterior, 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 anterior, and then posterior. And so when you have that sort of setting, the cell is going to be from that AB, which is at sort of like the first, it's like the 2-cell state, and that AB is the anterior to that. So it's like the first half of the the front half of the embryo. And then you get a couple of divisions where it's like towards the front, towards the right, towards the tail, then towards the head, then towards the tail. And that gives you a 3-dimensional location, which is here. And, this one here is CPAAP, which means it's posterior and anterior, but it's still located somewhere because the C sublineage divides and it moves to the right or left. So there's this spatial position you can assign to this. And so it's a 3-dimensional spatial position, and you can put all of these positions into a list. You put it into a neural network. You get this orientation map, which is, has these different directions, different amounts of movement. So this is a list of cells are pre-selected as their state cells via the cell neighbor determination model. So you have these 3D positions. You determine which ones are neighbors, being which ones are the nearest neighbors based on their X, Y, Z position. And then you might use like a Euclidean distance metric for that, just to determine which ones are closest. And then a list of cells are pre-selected as the state cells by the cell neighbor determination model. So these are the neighbors here kind of lined up in this row. It's like the 1-dimensional row of neighbors here. Their locations are concatenated, so you might select a number of 1-dimensional rows where you might have the X coordinates, the Y coordinates, and the Z coordinates, and the neighbors in those dimensions. And then their locations are concatenated and sent to the neural network, which is here. And the output action with the maximal probability is selected as the migration cell's next movement. So this is the movement direction, and this is, I guess, the probability, not the speed, but the probability that this is the right direction. And then you see the model, you have these cells that, they move in a certain direction, and then that determines the state, this next state of the ABM. So you're taking the positions and you're doing this recursively. So once these cells move, then you get new locations, and you do it all over, and you do it until it ends. So that's all I'm going to talk about with this paper. Any questions?
It's a nice paper.
Yeah,
I think I'll quote it.
Oh yeah. So a nice example of using MESA and some of these techniques. We've used MESA in my other group. We use MESA for a number of different types of models. It's pretty good to work with. It's Python extensible, which means you can plug other toolboxes into it. So you can combine MESA with reinforcement learning, which we've done actually. we've combined, agent-based models with active inference models and other things. So there are other things you can build in Python, other types of models, and you can make it into a hybrid model where you have an ABM and some other thing. And so that's really powerful because, the standard ABM model isn't necessarily, as useful as having other types of algorithms on top of it. Okay, the next thing I wanted to talk about today is, so there's been a lot of stuff out about simulating embryos. There's this set of papers, but there's also this sort of target article that came out in, I think it's Nature Methods. And it's an article on people building virtual embryos. And so this is a model that sort of predicts forces in an embryo. This is from the Turlier lab there in France. And they developed this technique where you can take microscopy images of an embryo and estimate the forces using, I guess it's some sort of finite element model, but I'm not sure how exactly they're doing it. The citation of the paper is in this paper. So, it allows you to estimate the forces across the entire embryo for all cells and cell interaction spaces. So this is where, you would do like in the last paper we saw that they had characterized the embryo, they characterized the cells moving around and the forces, and there are all sorts of interesting interactions there. In this case, they're just measuring the forces, and you can measure it over that, dynamically over the period when cells are dividing and migrating and getting a sense of what those are. So you're actually adding to the parameter space. So to develop computational embryo models takes experimental data, AI, and machine learning. The Turlier lab has developed a method to build a geometric map from imaging data. This is the 390 cell C-squared embryo, which we've worked with the data on that, but we haven't talked about it recently. So C-squarts are a great model for looking at embryogenesis. C-squarts have, there's data, not only is there data available, online, like we have with C.elegans, but it also has the same rules of C.elegans embryogenesis. That is that development is a deterministic thing, so we have this nomenclature. It's very different from C.elegans, but it's very descriptive in that the cells don't change their fate or they change their sort of, what they do over different embryos. It's very much like if I track like the B sublineage or the A sublineage, I know what the daughter cells will be and roughly where they'll be in the embryo. So I can know this information, and so that's an advantage for this type of embryo analysis and simulation. So this is the 396 cell C-squirt embryo, and C-squirts are interesting because they start out kind of in this deterministic mode, and then they have a number of different phenotypes throughout development, which are also interesting, but that's not the purview of embryo modeling. So how to build a virtual embryo. And so let's see, I'm going to go through some of these. It's basically just talking about different groups and doing things. We have the computational embryo models. This researcher, Berna Sozin, computational embryo models will be excellent companions to human embryo-like models and natural embryos themselves. She and her lab developed stem cell-based models called human extra embryoids. So this is where we get these computational models, which I showed in the last image, but we also have these other types of embryoid models. And so we've talked about some of the embryoid models in humans and in primates. And so the embryoid models are these organoid type models where you have stem cells and you do directed differentiation on the cells. And you develop different tissues. And so having these models, they're models in the sense that they're not actual embryos, but they're embryo-like models. And Morgan knows what I'm talking about from Saturday when we talked about some of these embryo-like or stem-like cells So they have different gene expression profiles than your typical embryo or stem cells, but they behave like them and they have very similar phenotypes, and so we can use them as models. So yeah, this is one example. So you can basically use computational models, or you can use embryo-like models to build and test theories, delineate new research questions, and make experimental design easier. And so it's very hard to do human embryo models or study development in sort of a setting where you can take an embryo and put it in a dish. You can do embryo models much easier, but still computational models are probably even easier than that. So we have to kind of think about how these kinds of models fit in. Computational embryos are good for a lot of things, not just looking at different things like forces or cell migration, but you can also do a lot of things with computational embryos. Like, for example, you can use it to assess how drugs affect embryos. You can look at model changes that could lead to congenital diseases or increase disease susceptibility. You can also help understand development in its different guises, and also use them as teaching tools. And you can do a lot of these things with the embryoid models as well. That's why we want to build embryoid models, because we want to understand cells in sort of the context of a multi-tissue embryo. So these models are getting more complex all the time.
I'm sorry. I'll just drink.
So these cells are getting more, or these models are getting more complex all the time. You have these integrated models, which include extra-embryonic tissue. Then there are less complex non-integrated embryo models that lack this type of extra-embryonic tissue. So you can build them at different levels of complexity and build them for different purposes. You don't need to build a full embryo model right off the bat. You can study different questions and include things as needed. So this, let's see if there's anything else in here that I wanted to cover. So this is basically one of the things I mentioned earlier about sort of soap bubbles and soap films. And, you can use that as sort of an analogy of the cells moving around in the embryo, dividing in different ways along different axes. So they talk about here, embryos are not bubbles, but surface tension shapes the geometry of both embryos and bubbles. So you can actually use bubbles as sort of like this meta model to describe the physics of embryos. So, bubbles are these spherical things that have to be packed into a larger film. The edges kind of bump against one another, so they have this sort of tiling that they achieve, this optimal tiling where they kind of fit in between one another. And you can see there's actually a part of an area of mathematics called soap bubble theory, where they kind of use soap bubbles as a model for sort of spatial optimization and so forth. So bubbles and embryos have a lot of similarities, so we can use some of those sort of insights, but we don't want to think that one is equivalent to the other. And so they kind of talk about here where you can integrate AI into these types of computational embryos. So they talk about this platform here from this group, one of the groups that they're talking about. They're talking about integrating different types of data into the model, so you can look at things like tension inference. So this is, I guess this is the Turlier lab where they are looking at these 4-dimensional maps of tension. And you can characterize cell and tissue mechanics theoretically and experimentally. You can also you can extract fluorescence from these models, so you can get a sense of gene expression. You can also integrate spatially resolved single-cell RNA-seq data. So this is all kind of part. You can do these different types of things. The platform that integrates AI trained on experimental data, biophysical models, and experimental data will be released soon. So they're releasing a model, an AI-driven model on some of this as well. One module handles forward modeling for simulating with modeling parameters. Another is for inverse modeling and uses parameters such as data extracted from micrographs. So this is where you have the forward modeling, which actually makes predictions, and the inverse modeling, which sort of gets data from a source and is, sort of things that you can infer things with. So you have prediction and inference with these different types of forward and backward modeling techniques. And then, they kind of talk about extracting cellular geometry from micrographs. There are different types of challenges that you have here, but that's part of the game, I guess. Yeah. So I wanted to go on to the other two papers in this group. So this first one is this is from the Turlier group. This is on embryo mechanics and the work that they're doing there. So they have embryo mechanics cartography, inference of 3D force atlases from fluorescence microscopy. This is kind of talking about how the methods behind these maps that they're building. So I'll just read through the abstract and then I'll move to the other paper. So this first, the abstract reads, tissue morphogenesis results from a tight interplay between gene expression, biochemical signaling, and mechanics. The sequencing methods allow the generation of cell-resolved spatiotemporal maps of gene expression. Creating similar maps of cell mechanics in three-dimensional developing tissues has remained a real challenge. So it's, kind of getting this sequencing data. You can get spatiotemporal distinctions, and we can do this for single cells, but creating similar maps of cell mechanics is harder. I would argue, a lot of the next-gen sequencing maps are not particularly insightful, so that may be the distance here. But exploiting the foam-like arrangements of cells. So this is where they compare the arrangement of cells to a foam. We propose a robust end-to-end computational method called Embryo. So this is a foam embryo, or like a foam-like embryo, to infer spatiotemporal atlases of cellular forces from fluorescent microscopy images of cell membranes. So they're segmenting the membranes and they're looking at interactions between the membranes. Our method generates precise 3D meshes of cells' geometry and successively predicts relative cell surface tensions and pressures. We validate it with 3D foam simulations, study its noise sensitivity, and prove its biological relevance in mouse, Ascidian, which is the C-squirt model, and worm embryos. That's for C.elegans. So yeah, they have these, I guess, they're in the paper. 3D force inference allows us to recover mechanical features identified previously, but also predicts new ones, unveiling potential new insights on the spatiotemporal regulation of cell mechanics and developing embryos. Our code is freely available and paves the way for unraveling the unknown mechanochemical feedbacks that control embryo and tissue morphogenesis. So if you look at this, this is the multi-material mesh generation algorithm. They walk through the algorithm. They basically, try to extract the membranes, so they're not really interested in the cell centroids or any of the contents of the cell. They're just interested in the edges of the cell or the membranes. And then they use a Delaunay tessellation for both the cells and the cell environment. They use watershed clustering on a dual Voronoi diagram, so they kind of build a model of the cell, the morphology, and the edges of the cell, and define those as the membrane. And then they have this multi-material mesh by a cluster border extraction, so they extract out the membranes. And so they're extracting out the background, they're extracting the membranes in the foreground. And then they have this virtual model here, which is our two cells that have an interface with each other. And then they benchmark this on some error and then angle values. So they actually characterize the different modes of interaction.
They're lucky that the actin in the cell actually is on the exterior. Like it tends to go toward the edges of the cell.
Yeah.
So this might be tracking where the actin lies in the cell rather than the cell membrane, which doesn't contribute as much as the actin does towards the cell.
Oh yeah.
Mechanical. I don't know.
Yeah, I know what you mean. It's like you have these different components of the membrane and the edge of the cell or the body of the cell that are interacting and giving it shape. So they're just extracting it from an image so they don't see they're not accounting for the three-dimensional depth of the structure or the edges of the cell or whatever.
Yeah, well, the cell mechanics is determined a lot by its actomyosin.
Right.
Which is on the exterior, like it tends to go toward the exterior of the cell. I don't know why.
Yeah.
Do you know why?
I don't know.
No? Okay. Well, it's
maybe that's where it's most useful. It doesn't really reason, but yeah. So I mean, So I guess their ideas is building these spherical models that sort of gauge where the interaction points are. And again, it's not like we don't have everything in there, and this may be minimal useful or maybe more useful. I'm not really sure, but they're able to do that for the. So this is just the segmentation step in any case. They're going to then simulate these interactions using different parameters. So they're going to separate out like the actual cell structure with sort of these interaction parameters that tell each cell that you're interacting at a certain force. These are the forces that we expect. So there's a surface tension you can map across the cell or the embryo surface. So this is a series of cells in an embryo. The surface tension is higher towards the bottom of the embryo and lowest at the top at the dorsal section versus the ventral section, and then there are intermediate tensions in the middle. And then pressure it's hard to see the colors, but it seems like there are lighter patches of pressure and darker patches of pressure. And then, of course, you have these other types of things here. So this is actually in the Ascidian embryo. This is the 64-cell Ascidian embryo. So the Ascidian embryo at this stage is pretty symmetrical. So, it sort of controls for asymmetries in shape. But you see these cells that have, this sort of structure, and you can measure the tension and the pressure. And then G, H, and I are down here. So G is an exploded view of the surface tension map. So this is where we have an E, the surface tension map, and G, it's exploded. So all the cells are kind of exploded out, and you can see the sort of the cells floating in space, and so you can see that. This illustrates cell-cell contact tensions within the embryo. So it shows like where on these edges they have different tensions. So in this edge, there's a higher tension than on this other face of this cell. So you can see that there are junctions where there's a high surface tension versus not. In H, you have force graph representation of the mechanical atlas. So this is a force graph network where you have the forces mapped to these interaction. So the nodes are the cells, and the edges of this network are the points of interaction. So again, you can see that these edges, these faces of individual cells have higher surface tension. This corresponds with this edge here in red. You can see it. So there are edges that are red, which means very high surface tension, and there are edges that are blue, which mean low surface tension. So this is almost like not quite a tensegrity network, but it gives us some information that might be useful there. See how those tensions are being regulated. Any thoughts about that, Susan?
Yeah, it looks like a tensegrity network. I wouldn't have gone with surface tension on this, but Yeah, I don't know. I'll certainly read this.
Yeah, yeah. And then I here is the 3D stress eigenvalue representation corresponding to stress tensors calculated per cell. So this is where you have these stress tensors in the same structure. So this is they kind of go through for Ascidian and for C.elegans. Let's see if they have like a comparison.
I would just say surface tension is important.
Yeah, yeah,
for sure.
yeah. So they actually show that topology changes as a measure of inference robustness. This is showing these unstable junctions between cells. So, if you think about like a soap bubble, they sort of smush together and they have these interaction points. And then as the soaps burst, they kind of come together. And so you could have, like in this case, cell death, where the cell dies and the other cells kind of take up that space and come together. Sometimes you have unstable junctions, so it could be like a cell dying, or it could be like cells are trying to move against one another. They're trying to make those active movements. And in that case, you get these displacements along the boundary, so the topology can change when you have unstable junctions. So at these junctions between cells, in this case, you have 3 cells that have a junction. They form a junction. They have these components of Y, and there are these forces in these different directions. And so when these are unstable, you can get these shifts in topology. And so that's what we see here. And then you can have that.
Oh, go ahead. Trijunction disappears.
Yeah, trijunctions, right?
Yeah, well, it's a construct in the cell, in cells and tissue.
Yeah.
It's separate. It's yeah, they're interesting. Did I send you any papers on trijunction?
Yeah, you did. We actually talked about it too. Yeah.
Okay. Good.
And so and then this is, of course, in the Ascidian cell here. So this is where you have something that's a little bit more sort of elongated, like what you would find in, just kind of showing the polarity, anterior, posterior, medial, lateral, then in both anatomical dimensions, what some of these forces look like. Okay, and then there's some references here. I don't know how useful they are. I don't know what kind of they have, if they have their code released or what they're actually working on with this. They actually have some tools. It looks like they kind of mentioned that in the discussion what those tools are. But there are ways you can display these morphologies and forces in multiple ways. So you can build force graphs and things like that. So there are actually tools attached to this. And so I don't know where those are exactly, but I guess we can find out in the work here. And then finally, this one here. This is the mechanochemical model recapitulating distinct vertebrate gastrulation modes. This is where they build a mechanochemical model, and so this is a virtual embryo. Gastrulation is a critical event in vertebrate morphogenesis, driven by cellular processes and characterized by coordinated multicellular movements that form robust morphological structures. How these structures emerge in a developing organism and vary across vertebrates remains unclear. So in the last part of that target paper, they talked about, will there be one single embryo model? And the answer, I would think, would be no, because embryos are extremely diverse. We have different types of embryos, different types of modes of development across them. We know that like development has like unified modes, like a lot of embryos have gastrulation and other types of stages and types of characterized movements, but they vary in how they're executed. So I don't know if we could have a single embryo model. So we want to know, though, at the same time we want to know that we can simulate these across different types of morphological structures. So to be able to have this kind of maybe a module that you can put into different embryo models to say this is like gastrulation would be, really nice. How these structures emerge in a developing organism and vary across vertebrates remains unclear. Inspired by experiments on the chick, so this is in chick embryos, we derive a theoretical framework that couples actomyosin activity to tissue flow and provides a basis for the dynamics of gastrulation morphologies. Our model predicts the onset of development, observed experimental patterns of wild type, and perturbations of chick gastrulation. So this is where we have the wild type and different perturbations of chick gastrulation. So it's not just the standard gastrulation mode as a spontaneous instability of a uniform state. So a lot of times you'll have mutations in the process, or you'll have in some, not in chick embryo, but you'll have defined mutants where you can see these effects of the perturbations of this wild type genotype. And so they characterize this as a spontaneous instability. So it's interesting. Varying the initial conditions and a parameter in our model allows us to recapitulate the phase space of gastrulation morphologies seen across vertebrates. So they're able to take this phase space, they're able to build a phase space for all different vertebrate species and for this simple gastrulation morphology. And this is consistent with experimental observations. And there's an accompanying paper here I'm not going to talk about, but they did some experimental observations and they match. Altogether, this suggests that the early embryonic self-organization follows from a minimal predictive theory of mechanosensitive flows. So this is sort of this active matter type approach. And so they talk about gastrulation, they talk about this process. So gastrulation is this essential, highly conserved process in the development of all vertebrate embryos, with the chick being an extensively studied model. During gastrulation, the chick transforms a layer of epithelial cells into a layered structure of three major embryonic tissues. At the moment of egg laying, the chick embryo contains around 600,000 cells organized in a central concentric epiblast that will give rise to the embryo proper, surrounded by a ring of extraembryonic tissue. So this is like when you crack an egg and you have the yolk and everything. And then during the first few hours of development, signals from the extraembryonic tissues, the epiblast, and the developing hypoblast induce mesendoderm precursor cells located in a sickle-shaped domain at the posterior end of the epiblast. So this just talks about some of these, the dynamics here. We have these directed cell intercalations that result in a contraction of the mesendoderm tissue towards the central midline, followed by an extension in anterior direction, forming the primitive streak, which is going to be important for later structure. And so there are all these processes going on. But the important thing to remember is that we have these force-generating processes that drive tissue flows. And so there are three that they define. The first is the outward migration of the cells. Two is the active intercalation of the mesendoderm precursors generating forces. And then three is the ingression of mesendoderm cells in the streak, which attracts cells towards the streak and drives out the plane motion. So that's basically like all these different flows are driving this process. And so the variation of tissue flows and morphology seen during gastrulation across vertebrates, and they, go all across vertebrates. So we're talking about fishes, amphibians, and reptiles to amniotes. And these are due to changes in embryo geometry and the relative contribution of these three force-generating processes. So that's what we have. And so this is a sort of an image of what we have here. This is phosphomyosin content for these different structures. We have the sickle structure, we have this HH2 phase, and then we have the primitive streak that forms from that. So it's kind of moving inward along the edges. There's pushing outward in the middle, and we're going from the sickle to this intermediate structure to this primitive streak, which is reinforced laterally inward. So this is where we get these flows, and you can see the force maps here, and that's that whole process. So active.
That's nice. That's very nice. I haven't seen someone describe the formation of the primitive streak before like
Okay.
this.
Oh
That's
yeah,
very nice.
yeah. And then yeah, so this is all driven by active myosin cables, and so you can see that they have this set up. Susan says very nice. Yeah. Yeah, and so that's in chick embryo, but they're going to then generalize that outward to other models. And so our model of the sheet-like embryo is assumed to be a compressible fluid to accommodate cell ingression. This implies that we need a continuity law that accounts for flow divergence or convergence. So they use this linear model, and this is kind of talking about some of the dynamics of the linear model here. Fluid bulk viscosity is characterized by C -1, while P0 characterizes the effect of an active cell ingression and can be thought of as the ratio between the isotropic and anisotropic active stress. So they have this more in the methods, but basically they're modeling this. Then we may write the equations for local momentum balance of the planar velocity field, which is v x, y time, along with equations for the orientational and intensity dynamics of myosin cables. So that's these two here, variations on v, this velocity field, and here are the equations. So this is what they're working with. So they have, they've laid this out in terms of a mathematical model. And this is where they kind of go through gastrulation, the dynamics of gastrulation. So they have the sickle, and then it goes to this primitive streak. So they've characterized these dynamics. So this is, let's see, we have D. Let's look at D here. D is model-based DOA at T0 and the attractor at TF. So this is T0 where you have the sickle. TF is where you have the primitive streak. So they're modeling between those two. Movie 2, which is the movie of this, which is in the supplemental materials, shows the time evolution of the relevant model-based Eulerian and Lagrangian fields. Eulerian, Lagrangian fields, same as D from the experimental velocity, that's E. So we're looking at here, model DOA, model attractor. This is experimental DOA, experimental attractor. So basically, we have a model, which is the mathematical model, and then we have experimental observations. So they match up pretty well. Okay. So this kind of goes through. They're using FTLE models, which are finite time Lyapunov exponents. So this is where you model from one time point to another, and you look at the trajectory between those time points, and you do some Lagrangian stuff to it, and it's good stuff. So yeah, this is just more about the developmental perturbations, so we can actually perturb this in different ways. We can look at model and experiment, and we can actually match those two together. This is now the evolutionary transition in these patterns. So we have, again, we modeled this in in the chick embryo, but we can extend this out to other vertebrates. So we have fishes, frogs, lizard and amphibian, and a chick. Basically characterize this across these different taxa. And so you have an initial condition, which is the sickle, and then you have this ancestral condition, which is actually this non-sickle, it's the spherical structure. So basically, they give this as the initial condition for our model. This is the ancestral condition. So this is at the sort of the basal part of the phylogeny. And then as you sort of get, when you have evolution, you have different types of initial conditions. So this ancestral condition, you see this in lizards and amphibians and fish. The spherical thing with the sickle, you see that as the derived condition in lizards and amphibians and chicks. And so that's what they're looking at here. And then, so they're looking at the different ways in which this primitive streak forms here. And then they're looking at the cell ingression process, lizards and amphibians and fishes. So they're making that connection between two parts of the vertebrate phylogeny. And then they're looking at the experimental forces, and then they're looking at this computational model as an attractor, and that's what they get. So they have this, they're looking at changes in the critical morphogenetic parameter P0. This is the isotropic to anisotropic active stress ratio, and regulates the amount of active cell ingression in EMT, and then the initial conditions MT0, which is the extent of the mesendoderm precursor territory. And this allows us to recapitulate these events in evolution, largely this event between going from this ancestral condition to the reduced mesendoderm. So the reduced mesendoderm is the crescent, and then it's, it has these same dynamics though, which is the point.
Yeah, well, there's the where Richard Gordon's waves
Oh yeah.
happen. That's the
Okay, so that, and so what did we have in the chat? Yeah, drop links to the papers too. That would be great. I'll put the papers in the DevoWorm Slack, but I'll also send them out in email form to Susan because I know she's not on Slack. That's
well, I'm on Slack, but I don't know how to use it.
Oh well,
being too much of a Luddite.
So yeah, it's we'll just send an email. Yeah. So that's good. So that's what and then that the first paper. I think there's some other papers in that issue of Nature Methods that are interesting as well. So I didn't cover those, but and then that last paper we talked about, they had an experimental companion, which I also didn't talk about, but.
Okay, so there's more.
Yeah.
Okay, I can maybe have a look. I need to get my stuff just finalized. Whatever I've done.
Yeah.
That's about it.
Okay. Well, great. Well, thanks for attending the meeting.
I'm here still.
Yeah, no. Any other comments or questions before we go?
No, you don't know how to run COMSOL, so.
Not really.
I haven't met anybody except COMSOL employees that know how to run COMSOL, so. It's just, that's a problem.
Yeah. All right. Have a good week. Good luck.
Thanks.
Yeah. Thanks.
Yeah.
Bye.